Hi everyone, I'm gonna present you the course of Microscopy Experiment from the African Bioimaging Consortium, ABIC, that's strengthening the microscopy community in Africa. In this presentation, the types of experimental questions which can be addressed with microscopy will be discussed. The different stage of a microscopy experiment will be repetitively mentioned while showcasing examples of typical microscopy experiments. What type of experimental question microscopy can best help you answer? The stage of microscopy experiments, experimental question hypothesis, planning approach, acquisition of data, processing and analysis of data, an example of typical microscopy experiment. In the introductory level curriculum of ABIC, the previous lecture covered topic as introduction to light microscopy technique, fluorescence microscopy, sample preparation for microscopy, and microscopy images analysis. Microscopy allows us to very minute particles from bacteria and virus and to the cells. We need to realize that we can get a lot of information and data from microscopy images. Often microscopy is seen as a technique only to acquire pretty pictures, but it's not the case. We don't have to limit that. We can actually obtain data from microscopy experiments. We can get qualitative and quantitative data from the imaging experiments. What is information we can get from the qualitative microscopy experiments? This type of data is based on observation. For example, looking cell form movement, how those cells move, how the filament arrange, structures, or trafficking. Microscopy allows visualization of the sample instead of only a quantification of the sample, which is the case for Western blot. With microscopy, we can visualize arrangement and localization. One can visualize shape and structures, whether an organism is rod-shaped or circular. One can detect whether the cells are dead or stressed. They are all qualitative examples. However, many of these examples can actually be quantified as well. In previous decades, it was sufficient to publish observation and qualitative data. In more modern research, reviewers require quantification of imaging information to prove significant differences between controls and the experimental question. So the quantitative microscopy experiments. So instead of visualized description of the image, questions has, so how many, the count of cells, nuclei, organelles can be addressed. One can quantify how big, the length, area, volume, rates. Even parameters related to the shape, such as aspect ratio, circularity, can now be quantified. In live cell experiments, one can report on distance traveled, velocity at which the cell migrated, on the directionality of the movements. With the development of the fluorescence technique, the intensity can be measured, providing a means to quantify the distribution at levels of the molecule labeled. To plan or design a microscopy experiment, it's important to know the different type of microscope. So first, you have the bright field microscope, mostly used in our histology labs, using the natural pigments of the sample or staining with colorimetric dyes. The fast contrast, this is a technique relies on the image of the sample changing with different phases of light illumination. This type of microscopy provides more contrast than what is available in bright field microscope. This is very nice for live cells experiment and is often used in cell cultures. The dark field microscopy provides contrast in algae, planktons, diatoms, insect fibers, hairs, yeast, and protozoa. You have another optical microscopy technique like DIC and polarized. So sample like teeth, bones, or straight muscles can be visualized with this method, parallelized microscopy. So depending of your sample and the research question, you can choose one of these techniques that will show the results the best. You have also the stereomicroscopy is useful for dissection, looking at bigger, thicker sample. And you have the wide field and microscope for fluorescence sample to visualize subcellular localization and get more resolution. On the next slide, microscopy experiment will be discussed in a research context. There are 5 categories of example. The first one is an example of location distribution, looking at the location of a protein, for example, how it is distributed in cell 1. We also have an example of object shape analysis rather than just describing what is visualized. We can actually data from such experiment for a better comparison. Another example is an object tracking, where the way an object moves can be converted to number for reporting and comparison. One can also obtain quantifiable data from localized signal quantification. One can compare signal intensity in different parts of the cells or sample, from example, in the nucleus versus the cytoplasm, but also compare it with other sample. And the last will be in object counting. So this is the different stage of the experimental plan. So the first is, what is your question? Is it a qualitative or quantitative question? What parameters are you interested in? Big, far, many, how fast? The second, in which model will this experiment take place? In tissue or cells? Are you interested in the organization of cells? Does it have to be a live or a large sample? Are you looking at movement, sample preparation, consideration? Which labeling strategy can be used to visualize the content of interest? What is required for the cells to stay alive or healthy? You need to plan what control you need. This is very, very important. The third is what microscope you need. A confocal might seem like the best option, but it's not at all case the best option. Sometimes a wide field microscope is adequate, while bright field microscopy could be the most appropriate method in other cases. What magnification is required? High resolution is not always going to be the most appropriate aspect. A lower magnification, which does not need to be high resolution, could be better. You also need to decide if you want to image 2D, 3D, or 4D. 4D is, for example, for time-lapse, if it's required. And finally, you need to determine what analysis should be done to answer to the original research question. Let's start by an example of distribution of a molecule. So we chose an example of the diabetes disease and the distribution of insulin of glucagon. So how applicable this example is where to you? How translatable the way to your research? We know in diabetes, we know the human body, the organ, the pancreas. There are alpha cells produce glucagon and beta cells produce insulin. And we know that both of these cells work in concert to regulate the amount of glucose we have in the blood when this is in balance. So imbalance in blood glucose level are usually what give rise to diabetes, characterized by abnormal high blood glucose level. So researchers studying this condition would like to know the percentage occupation of beta cells and alpha cells in the islet of Langerhans to identify the diabetic condition. So for that, the question is, What is the percentage distribution of beta and alpha cells in the islet of Langerhans? How many cells are in the islet? How many cells in beta or alpha cells? To process, we obtain pancreatic tissue sample, including in healthy control, and stem them appropriately. So we fix there, we stain cells with the flushing that labeling the beta cells red in red, and the dab labeling alpha cell in brown, and the hematoxylin control stem cells all cell blue. After that, we can image our cells just with a transmitted light microscope to see the total and the aspect of your cells. So scientifically, we need value to this. We need some significant stage. So we need to filter the noise and all of that. We need to segment out. So now after, we can count cells in the image. After filtering, denoising, and segmentation, we can measure and we can have some plot that count all of cells per area, and after tell us the proportional cells alpha in your sample, for example, to have the location distribution of insulin and glucagon. Let's look at another example, object shape analysis. So now this is an example of sickle cell disease. So sickle cell disease is a genetic blood disorder that inherits its paths from parents to children. Babies are born with sickle cell disease when they inherit two abnormal genes. They inherit one from each parent. A baby who inherits one abnormal gene from one parent is a carrier. They will have the sickle cell trait. If the baby grows up to have a child with another carrier, then their baby might have the sickle cell disease. If sickle cell genes cause episodes where the body's red blue cells become rigid, inflexible, and misshaped, changing the disc shape to a curve shape that looks like a crescent moon or an old farm tool known as a sickle. These sickle cells tend to stick together here and block the flow of blood and oxygen to the body, causing pain in the extremity and back, infection in the skin and other areas, organ failures and other tissue damage, loss of eyesight, severe blood clots, and strokes. These complications tend to progress at adolescence and worsen during early adulthood, and often lead to early mortality. So how can we describe this condition based on this? How can we know? All right, so we can quantify microscopy to use the different sickle cells with the control and know the difference, because we know the difference of the shape in the sickle cells anemia. So it makes sense. We want to use microscopic quantitative to differentiate sickle cell patient from healthy control to characterize the different groups. So it makes sense to take a blood sample from sickle cell patient and to healthy control. After we perform it, we use just a transmitted light in a 400 magnification and the all immersion, and we can see very good the difference of shape. Here the round, circular, and here the incurved sickle cells. After that, we can also measure the aspect after filtering, the noising, background, subtraction. we can have the diameters and all these parameters, if it's normal or circular. This is an example for the literature. So the affect cells change the shape. So from circular to sickle shape due to the presence of typical hemoglobin called hemoglobin S. So they did a processing technique to find and measure the different sickle cells. So the elongation affect cells are identified using imaging processing techniques, the shape descriptor, which are the vital features used to identify any shape in an image. To detect the sickle cells properly in the blood smear image, the image is converted here in a binary in order to find the boundaries of the cells in the image. In the primary image, each normal cell will have a hole here. Since the normal red-blue cells have the shape of a donut that have been pressed in the middle slightly, the boundaries of all the cells in the image is traced, and the holes inside each cell are filled. Each cell is considered as a connected component for which the eccentricity is calculated. The eccentricity of the shape descriptor, which is the ratio of the major axis and minor axis of the connected component, if the eccentricity is greater than the threshold, the cells are identified as sickle cells. The second example of shape is about mitochondrial networks. So mitochondria is responsible for the energy regeneration generation in the cell and is polarized across the membrane to allow electron transfer. The healthy mitochondrial network is in constant equilibrium with fission, fragmentation versus fusion happen in other parts. One of the functions of this process is mitochondrial quality control. Damaged mitochondrial strands are removed and depolarized to prevent them from fusion to the network again in preparation for the degradation. The equilibrium may change depending on the metabolic demands on the cells and a better understanding of these dynamics and their effects will provide insight to possible treatment strategy during the phase of neurodegeneration. So to know the shape analysis, we want to see the question, how does the mitochondrial morphology relate to cell stress or survival? So we grow cells to about 70% circumferences and administrate staining with a mitotracker red and apply different drugs, for example, bafilomycin. Now we can just see in confocal fluorescence microscopy in fixed or live cells the difference of shape. And after, we can measure the different parameters like branching, length of individual strand, fission events, fusion events, or circularity. So an example how data can be displayed and published. So in this publication, the autophagy modulating drugs, there is the control and different drugs like hydroxychloroquine sulfate here, as well as here is a chemotherapeutic temozolomide and also the rapamycin and the baphyllomycin. Donc, they coordinate autophagy modulation in mitochondrial network connectivity. Here, we can see the maximal intensity projection of gliomal cells stained with mitotracker red with the magnification 60. And after here, we have the means of the different gradients of with a different drug. We take the same area here. We can report the same area in different conditions. And we can have the connectivity index here between the different clusters. So in order to relate mitochondrial network dynamic through their topology, the connectivity between mitochondrial subunit was quantified in terms of the degree of scaling within disease networks and the amount of overlap between membranes. So scaling invariance was calculated by fitting to a power law relationship to scatter plots of the outer parameters and inner area with regard area of each network cluster with regard to the pure network properties of the mitochondrial network. The ratio of the total number of vertices to the number of connected clusters for a given graph was assessed, providing a connectivity index. So this is an example you can see in the report. That is very important to quantify the shape of our sample, to know here the different effects of the drugs. Okay, so let's look at the object tracking. So it's a quite common asset actually, like in cancer study, where they want to look at movement. So they want to look at migration of cells. So tracking the velocity or migratory behavior of cancer cells can help you to determine activation state, proliferation at metastatic activities, identify the drug that activate or inhibit signal. It's very important. So how we can do? So the question is, how fast do the cells move? Do cells change morphology when migrating, when contacting other cells? So we put on the petri dish the cells growing. In the middle, we do a cell depression with a scratch assay. After that, you have a hole here, an area, and in each side, you have the cells. So we can follow the recouvrement of this area in fluorescence, just in white field fluorescence with a fast contrast. And after, we can analyze the object tracking, displacement, chase parameters, and velocity of the cells. Why it's important? Because the question is, what is the area at the open area? How fast does it change? How can analyze the shape change? And are certain proteins upregulated or downregulated during migration? So take this example from the literature. So it's a bit complicated, so not to focus so much on integrated details. We just focus and now look at the displacement. So here we can see the shape of the movements. Here, for example, at the beginning, 6 hours and 12 hours, we can see that the area is reduced. And it's the same here, but the difference is the shape. Okay, it's not regular. Donc, there is, in these papers, there is an image processing to measure this area and the velocity. So the processing consists of noise filtration to enhance the cell contours or contrast, and then thresholding and binary mask rendering. So the first here, the software process is consequently each input grayscale image and applies image correction procedure where noise filtering and histogram reassignment are performed. Then the script applies a threshold on the filtering grayscale image to convert it into a binary form for correction and gap detection procedure. The obtained binary masks are then collected into a sequence of wound contour and area count here. So now what we can see, we can perfectly straight is that line or not, how is fast is closing is depending of the different treatment. We can see the different histogram, we can see if it's increase or decrease everything. So it's the shape, the movement of this is very important to know the effect of the drug, of how the cells are moving. Let's look at the localized signal quantification. So in this example, this is the example of prostate cancer. Nuclear transportation in cancer promote cancer cell multiplication. We have testosterone bind to the receptor. After androgen receptor translocate to the nucleus, and transcriptional activity of the androgenic receptor. It has been found that castration could prevent further development of the cancer. But however, in castration-resistant prostate cancer, androgenous receptor is found to still translocate to the nucleus and promote further development of the tumor. So we need treatment strategy aimed to block this translocation in prostate cancer. So they are trying to understand why is it still translating right? Can we stop this translocation? Obviously, they want to see movement and the localized signal concentration. How much of the androgen receptor in the nucleus compare to the cytoplasm? So this is our question. So to answer to this question, we can put cancer cells with transfected with an androgen receptor targeting with yellow FP, and we can do treatment with different hormones like testosterone, for example. And we can see it, for example, in ethanol control that it is cytoplasmic, and in other treatment, it's nucleus. And we imaging with confocal microscopy, for example, at eye magnification, it's we had a very good resolution. So after, how we can quantify these parameters? So we need to look the ratio between the cytoplasm versus the nuclear signal, the fluorescence intensity of the androgenous receptor and the cytoplasm and the nucleus respectively, and change all the parameters over time. And how, with that, we can answer to the different question of to see the effect of the treatment. So in this publication, this is an example of a quantification of different nuclear translocation published. So this is the size variance of the androgen receptors that's actually causing this nucleus androgenin. So they had the full and androgen receptor here transact with GFP. And then they had some variants, and so it has transferred it, and they see as a different distribution. So now this is the object counts. Let's look at so popular ones. It's an example. So a popular experiment that we use is to measure the viability of your cells. So it's important to count the cells, for example. So counting the viability of the cells is very important for a lot of application. For example, for testing antibiotic in bacteria or antimicrobial agents that should kill microbes, to testing pharmaceutical agents and testing their toxicity. Example also for the protective effect of antioxidant and natural products such as the South African rubius can be assessed on several cell types. So how we can do to count the viability assay? So we use the propydium iodide. So it is a membrane impermeable dye. So it can, when the cell is dying, it can reach nucleus and we have a positive signal in dead cells. So the question is, does the treatment cause a cell death or cell survival? So what is the ratio of live cells versus dead cells in the sample? So to obtain that, we obtain a microbial or cellular sample and stain with viability dye such as propydium iodide. And we need another staining to determine the total number of cells, like X, for example. For the live cells, for example, we need a control, and we need a dead control for positive signal. We can image this in live cell imaging or in confocal, in fixed cells, or in wide field fluorescence. It was very good. And after, we can count the cells to do the ratio between the X cells and the number of cells, total number, and the dead cells, staining with the PI. So it's very important because now we can see if the new drug is toxic and it's interesting. So we can do a test of viability and see if my drug is still or not my cells, for example, in the cancer cell on other bacteria or other disease. So this is an example for the literature. So in this example, this is a biomedical purposes, especially in vivo application like drugs delivery system. So they want to see the toxicity because in the drug delivery system, the toxicity is a critical factor which ultimately determines the suitability of a material. So in this, they use an essential, the polymer, and want to see if it causes a not effect. So the cell toxicity is measured in terms of cell death to see or not if is the study of apoptotic and necrotic cell death provide detailed information regarding the cell viability and potential cytotoxic effect. So the first here, this is a positive control. So in the positive control, the cells were treated with a known membrane permeabilizer. This is a ratio 1:1 methanol acetone mixtures. This serves as a positive control for cell death to identify that there is here in cell death with the nucleus stain in red. And here for this is the cell treated with the block copolymer, and we can see, so it's stained with the red staining of the nucleus and is not detected. So it's that which indicated that the cell membrane were still intact. So it's very important because now we can see that this pollen is not toxic to our cells. Okay, so now we are coming to a close to summarize our microscopy experiments. So the first, it was, what is your question? It needs to be a valid, clear, and smart question. What do you want to achieve from these experiments? How do you want to go about it? After your experiment, what is your parameters you need? It's very important. And for your experiment planning, what is your sample, the treatment, the transfection you need, your staining? You see in our example that is not the same for example, a receptor, live cells or stem cells. If you want confocal, if you want a fluorescence or just white field or bright field or contrast field, it's not the same. Also, for the acquisition of image, you have to took the consideration of what type of microscope you need. So what you need to use, not just a microscope, but see the available. Would that microscope answer your question? Will it solve your problem? The magnification. And also another thing is very important is the data analysis and interpretation. You might need to consider modification, your data processing, how I analyze the data to get the answer of your question. How are you going to represent your data that it will make sense to somebody that is reading your paper? It's very important to find your pipeline to all your experiment and data analysis. Okay, then so that's all. So thank you, and thank you for your attention.